Hi, and welcome to another episode of Tom Kennedy Science, and I'm, of course, your host, Dr. Tom Kennedy. Now, today is my fourth lecture in a series on photosynthesis, and what we're going to talk about today is the Calvin cycle. And the Calvin cycle is a series of metabolic reactions. These are chemical reactions that are going to fix carbon dioxide into organic molecules. So let's start with the big picture. You know me, I like to always dive in with the big picture. So what are we doing here? Okay, so photosynthesis in general, photo means light, synth means put together, or putting together by a light. So we're going to take some carbon dioxide, we're going to take some water, and we're going to take the energy in sunlight, and we're going to use that to make an organic molecule like glucose, and in the process, we're going to release oxygen, okay? Now, we know that this is a redox reaction, okay? Reduction, oxidation. So what's going to happen is we're going to take carbon dioxide out of the atmosphere, and that's a very oxidized form. If you notice, the electrons are away from carbon, they're closer to the oxygen. So we are going to reduce carbon into an organic molecule, and that means we need, of course, a source of electrons. So where would they come from? Well, if you remember, they come from water. I know. It's just so befuddling to me that photosynthesis is capable of ripping electrons away from oxygen and the protons too, right? And by doing so, the process releases molecular oxygen, which, as you well know from my first lecture on this series, that revolutionized the planet, paving the way for multicellular life like you and me. Okay, so that happens in the light reaction, right? We're going to split water, we're going to elevate those electrons in energy, and then we're going to send them over to the Calvin cycle via our electron carrier. Now, one more thing about this reaction. It's not only just a redox reaction, it is also an endergonic reaction. We know that endergonic reactions require energy, okay? That means that the products, which would be glucose and oxygen, they store more energy than the reactants, carbon dioxide and water. So we need a source of energy for the Calvin cycle. And that source of energy, of course, ultimately is coming from the sun. And the light reaction is basically making the electromagnetic energy available as chemical energy, right? So that's where our high energy electrons come into play and ATP. So here it is. This is the Calvin cycle. You can see that the light energy is coming from the sun, that the light reaction captured this energy in the chlorophyll molecules, and then we transform kinetic energy into potential energy stored in the bonds of ATP, and also in those high energy electrons that are going to be carried over to us by the electron carrier NADPH. That's a very similar carrier to what we see in cellular respiration. This one just has an extra phosphate group attached to it. So here's our overall view of photosynthesis, right? You can see sunlight's coming in, it's hitting the photosystems, and basically in photosystem 2, we're going to split water. That's going to be our source of electrons, and it's going to create molecular oxygen. Then those electrons are going to be elevated in energy. They're going to run through an electron transport chain. And oh yeah, you know what's going to happen here? We're going to pump protons into the thylakoid space, and that's going to generate our ATP through chemiosmosis. All right, and then those electrons are going to be shuttled to photosystem 1, where they will be elevated back in energy again, and then they will be shuttled over to a large enzyme complex called NADP reductase. And then you can see on the bottom, there's where our NADPH is going to get reduced. And well, actually, NADP is going to get reduced to NADPH, and then we're going to form ATP. And that's going to be shuttled over to the Calvin cycle. And this makes sense, right? Because the Calvin cycle, we're going to take inorganic carbon, we're going to reduce it, right? You need your source of electrons. That's where NADP comes from. And also we need protons because, organic molecules have carbon and at least attached to one hydrogen. So you can see it's carrying hydrogen there as well. And it's also going to carry some extra energy in some forms of ATP. And this process is going to kick out some carbohydrates, specifically a 3-carbon sugar. I just want to be clear that the Calvin cycle itself, it actually doesn't make glucose, okay? It actually kicks out a 3-carbon compound. Now, electrons, remember, they link the light reaction to the Calvin cycle, okay? Now, the Calvin cycle. This is a series of chemical reactions, right? And now it has three phases. Let's break it down to what it's doing here. And the first one is carbon fixation. So whenever we talk about carbon fixation, what we're doing is we're taking carbon dioxide from the atmosphere, right? We're going to bolt it onto an organic molecule. And this is done by this enzyme called Rubisco. I know it sounds like Nabisco, doesn't it? Every time I've heard the word Nabisco, I always think of Rubisco and vice versa. And in fact, a few years ago, I saw the Book of Mormon, and the character called one of the other characters Nabisco, and about fell out of my seat because he could never remember her name. But I can tell you Rubisco, that stands for ribulose bisphosphate carboxylase oxygenase. Whew, that's a lot of words, isn't it? Ribulose bisphosphate carboxylase oxygenase. Rubisco, I'm glad we call it Rubisco and not the full term, right? But that word means something, and we will come back to what it means. Okay, so we fix carbon into an organic molecule. And as you know, the Calvin cycle, we have to reduce carbon dioxide into an organic form. We're adding electrons to it. So the third phase here is reduction, and this is where our NADPH is going to come into play. And then we have regeneration. So we have an acceptor, right? We have a carbon dioxide acceptor, and it's called RuBp, ribulose bisphosphate. Now, you probably have heard of ribulose, right? I mean, ribulose, ribose, RNA. Wait, ribose. That's a 5-carbon sugar. Bisphosphate, bi means 2. So ribulose bisphosphate is nothing more than a 5-carbon sugar with a phosphate group attached to each end of it. And that should immediately let you know a couple things. That molecule has a lot of potential energy, right? It's an activated intermediate, and it's got phosphate attached to it. Why do we need phosphates? Why do we need ATP? We're increasing the potential energy of these molecules, making the bonds easier to break. Okay, let's take a closer look here. There's the Calvin cycle. It's there's a few steps going on there. You know, a lot of times we look at a metabolic pathway, or we look at something complicated in general, and we're just like, oh gosh, how am I going to learn this? Okay, you know, we have a lot of us have learned this, and the reality is it takes some effort, right? Look at it a few times throughout the day. Follow the carbons. Where are the carbons coming in? Where is the NADPH coming in and reducing it? How are we regenerating our acceptor? Why do we have 3 carbons coming in? You know, in cellular respiration, we talked about acetyl coenzyme A bringing in, you know, an acetyl group, which is 2 carbons, and that's it. Well, the difference here is cellular respiration, we're just oxidizing organic molecules to carbon dioxide. Here, we are reducing carbon dioxide into organic molecules, right? And we're outputting a 3-carbon compound called G3P, or glyceraldehyde 3-phosphate. Okay, so if we're going to output 3 carbons, what do you need coming in? 3 carbons. So follow these around. Look at beginning, it's got the ball and stick model of just the carbons, and then you can actually see the molecular formulas next to it. I really like looking at the structural formulas. Do you know where all the carbons are? Do you know what those lines and those nodes are? Do you know where the hydrogens would be? Make sure you know all of that, right? Like, I mean, just look at the RuBp, the ribulose bisphosphate. You can see the phosphate groups on the end of it. You can see that it's got 5 carbon atoms. So once you start getting used to looking at it, it becomes less difficult to understand. Okay, so that's my study tip. You need to familiarize yourself with this by just kind of reviewing it several times a day for several days. So let's start with carbon fixation. So carbon fixation begins, you take a molecule of carbon dioxide, CO2, right? And we're going to use the enzyme Rubisco, ribulose bisphosphate carboxylase oxygenase. This is telling you something. Our acceptor is RuBp, ribulose bisphosphate. So it's a 5-carbon compound with basically 2 phosphates at either end of it, okay? Now, Rubisco is an enzyme, and that's the carboxylase. So what it's going to do is going to take carbon dioxide and stick it onto RuBp. And that's why it's called ribulose bisphosphate carboxylase. Because if you think about a carboxyl group, it's a carbon bound to 2 oxygens. Well, what is carbon dioxide? A carbon bound to 2 oxygens. So when you take carbon dioxide and break one of the covalent bonds to the oxygen and you use it to form a covalent bond to another carbon, you no longer have CO2, you have a carboxyl group attached to your 5-carbon sugar, and now in total you have a 6-carbon compound. Okay, and the reason why RuBp is phosphorylated, remember, one thing about doing endergonic reactions. How do we do an endergonic reaction? It requires a source of energy, right? So we have energy coupling. So we couple an exergonic reaction with an endergonic reaction. And one of the ways we do this is we use ATP, right, to phosphorylate an intermediate. And by phosphorylating our intermediate, we make it easier for those bonds to break by adding potential energy. So our RuBp is incredibly reactive, right? And it allows us to allows Rubisco to stick that carbon on there and form a carboxyl group. Now, once that 6-carbon compound is formed, it is incredibly unstable. It breaks apart almost immediately into 2 3-carbon compounds known as 3-PGA or 3-phosphoglycerate. Okay, so now we've got carbon fixation. All right. Well, remember, endergonic reaction requires a source of energy. So we're going to phosphorylate this intermediate once again. This is energy coupling, right? We're using the energy of an exergonic reaction to drive an endergonic reaction, which is the formation and the reduction of this molecule. So let's go to the reduction phase. So we phosphorylate it. Now we're going to reduce it. What do you need for reduction? Electrons. Who's carrying our electrons? NADPH. So this is where NADPH comes in and is going to start to reduce our 1,3-bisphosphoglycerate. Remember, we had a 3-phosphoglycerate. Now we've got a 1,3-phosphoglycerate because we added an ATP for it. This is a really reactive molecule, right? And now NADPH, we're going to oxidize that molecule and we're going to reduce 1,3-bisphosphoglycerate. Now we're going to reduce it to glyceraldehyde 3-phosphate. And one of those phosphate groups are actually going to pop off that molecule. Now at this point, one of those G3Ps, the glyceraldehyde 3-phosphate, that tells you it's on carbon number 3, is going to come out of the Calvin cycle, and then the cell will then use that, the plant cell will then use that to make glucose, all right? That's why 3 carbons come in at a time. Now the next step is what's called regeneration. Now in regeneration, we have to go from glyceraldehyde 3-phosphate, which is a 3-carbon compound. We now have to make ribulose bisphosphate, which is a 5-carbon compound. Okay, we're actually going to make 3 of the RuBps. That's a total of 15 carbons. Okay, that's why we have 5. Okay, we're going to use 5 of the G3Ps, the glyceraldehyde 3-phosphates. And through a series of chemical reactions that include, once again, a source of ATPs to phosphorylate those intermediates, we're going to regenerate ribulose bisphosphate. Wow, isn't that cool? Okay, that's the Calvin cycle. I haven't gone into every single chemical reaction here because I want you to focus on carbon fixation, carbon reduction, and then regeneration. It's these 3 phases to understand kind of what's happening and follow like why it comes in with 3 carbons at a time. Okay, that's important. Now, a couple other things you are probably wondering about. Now, some of you may know a little bit about glucose. Glucose has like 586 kilocalories per mole. That's a lot of energy. And many of you may know that ATP has 7.3 kilocalories a mole. Well, let's add up. How much have we used? Okay, so if you're kind of doing some math balance, you realize that ATP has 7.3 kilocalories a mole. And by the time you add up all the ATPs used here, there's like 18, right? We got 3 in the regeneration phase. We use 6 in the carbon fixation and reduction phase. Well, that's 9. And we need to double that because we've got glucose, which has 6 carbons, not just 3. That's 18. Well, 18 times 7.3 kilocalories a mole, that's just a little over 131 kilocalories a mole. Wait a second. Doesn't glucose have like 586 kilocalories a mole? Where does that other energy come from? High energy electrons. Most of the energy actually is carried over by the NADPH, and most of the energy is actually from the reduction by adding the electrons to it. So NADPH, those 6 NADPHs carry the remainder of the energy, like over 400 kilocalories a mole. So when you add it all up, those NADPHs are carrying almost 75 kilocalories a mole a piece. Wow, they have almost 10 times the amount of energy as a single ATP molecule. Pretty wild, isn't it? Okay, so there's the Calvin cycle. Now, what I want to do now, let's talk about Rubisco. I know, not Nabisco, Rubisco. This might be the most abundant enzyme on the entire planet because this enzyme is what fixes carbon dioxide into an organic molecule. It adds carbon dioxide to RuBp. Wow. Rubisco is ancient. I mean, it's been around probably since the beginning of photosynthesis. And we know this. What's interesting about Rubisco is that it evolved in an anoxic environment. It evolved in an environment that did not have free oxygen. And as a result, if you have low enough concentrations of carbon dioxide and high enough concentrations of oxygen, this enzyme will then begin to fix oxygen onto RuBp instead. And we'll talk about that later, but that's the source of photorespiration. And in that case, you're actually beginning to oxidize things. So that's a problem with Rubisco. It can actually fix oxygen, hence the name ribulose bisphosphate carboxylase. Carboxylase is when it's adding carbon dioxide to RuBp. The oxygenase is when it's undergoing photorespiration. When there's not enough carbon dioxide, it will actually add on oxygen to RuBp. Pretty crazy, isn't it? One other thing here. When Rubisco adds carbon dioxide to RuBp and it forms a carbon compound, that carbon compound, like I said, it just doesn't last. It's really, really unstable. And it immediately splits into 2,3 carbon compounds. And because it splits into 2,3 carbon compounds, we call that a C3 pathway. And that's going to be setting me up for the next lecture, C3, C4, and CAM pathways. I want you to be clear that every plant on the earth, no matter if it's CAM, C4, or C3, uses Rubisco to fix carbon dioxide into an organic molecule. So not only is it the most abundant enzyme on the planet, it's also one of the most important enzymes on the planet because it makes carbon available to life. Every carbon atom in you, hold on, let's go back here, is over 5 billion years old. It's been recycled over and over and over again. The carbon in you has been in rocks. It has been in oil. It has been in dinosaurs. And every single carbon atom in you was actually carbon dioxide not so long ago, fixed by Rubisco inside of a plant, and now it's inside of you. Just think about the journey that carbon is having. And carbon 12 and 13, those stable isotopes basically will last, 10 to the 35 years, which is way longer than the age of this universe and is basically immortal. They're going to have a long history. They're going to continue to keep going on. But every carbon atom in you was fixed inside of a plant using Rubisco. How cool is that? Okay, this has been another episode of Tom Kennedy Science.